Red Queen Hypothesis, ChatGPT said, ChatGPT. The Red Queen Hypothesis. The Red Queen Hypothesis is an evolutionary theory that proposes that organisms must continuously evolve to survive and reproduce in an environment where other evolving organisms are constantly competing. The term, Red Queen, comes from a character in Lewis Carroll's Through the Looking Glass, who tells Alice, it takes all the running you can do, to keep in the same place. In the context of evolution, this metaphor suggests that species must keep evolving at a certain rate just to maintain their relative fitness compared to other species in their ecosystem. 1. Overview of the Red Queen Hypothesis. The Red Queen Hypothesis was proposed by evolutionary biologists Van Vallen in 1973. It provides an explanation for why species undergo constant evolutionary changes. According to this hypothesis, evolutionary change is not necessarily aimed at improvement or increasing complexity but rather as a response to other species' evolutionary changes, such as competition, predation, and parasitism. The central idea is that in an ecosystem, the relative fitness of a species can only be maintained if it continually adapts to the pressures exerted by other organisms, including predators, competitors, and pathogens. Failure to do so would result in the decline or extinction of that species, similar to how the Red Queen had to keep running to stay in the same place. 2. Core ideas of the Red Queen Hypothesis. The key components of the Red Queen Hypothesis include constant evolutionary arms race. Evolutionary change in one species can drive adaptive changes in other species. For example, predators evolve better hunting strategies, which puts pressure on prey to evolve better defenses or evasion tactics. Fitness landscape. The concept suggests that a species' fitness is dynamic, not static. To maintain a stable position in the ecosystem, species need to keep evolving in response to changes in their environment or in other species' traits. Coevolution. As species evolve in response to each other, they are involved in a coevolutionary process, where one species' adaptation influences the evolutionary trajectory of another species, such as in the predator-prey relationship. Red Queen versus the Cheshire Cat. The Red Queens need to keep moving parallels the constant need for evolutionary change. The Cheshire Cat represents species that may evolve in a way that gives them a temporary advantage but might eventually reach a fitness peak, where further evolution is not necessary to maintain that advantage. 3. Ecological context of the Red Queen Hypothesis. In the natural world, the Red Queen Hypothesis applies primarily to ecosystems where species interact in complex ways. This includes predator-prey interactions. Predators evolve better strategies to capture prey, while prey evolves new defense mechanisms. This results in an ongoing cycle where both predator and prey continue to evolve in response to each other. Host-parasite dynamics. Parasites evolve new strategies to infect and exploit their hosts, while hosts evolve immune defenses to resist infection. Each evolutionary step by the parasite forces a counter-response from the host. Competition for resources. Species competing for the same resources, such as food or space, must continuously evolve to gain a competitive advantage. This can lead to shifts in traits such as body size, behavior, or reproductive strategies. Sexual selection. In sexual reproduction, individuals must compete for mates. Traits that make an individual more attractive to potential mates, e.g., bright colors or elaborate displays, can evolve as a result of sexual selection, but these traits can also attract predators, leading to a balancing act that drives evolutionary change. 4. Mechanisms of evolution in the Red Queen Hypothesis. The Red Queen Hypothesis operates through the mechanisms of natural selection, sexual selection, and coevolution. Natural selection. Species evolve traits that enhance their survival and reproduction. However, these traits may be influenced by the evolution of other species. For example, a predator's evolution of faster speed may lead to the evolution of quicker reflexes or camouflage in prey species. Sexual selection. Traits that are beneficial in attracting mates, e.g., bright feathers in birds may increase reproductive success. However, these traits can also make individuals more vulnerable to predators. The Red Queen Hypothesis suggests that the coevolution of sexual traits and predator avoidance strategies results in a continuous evolutionary struggle. Coevolution. Coevolution occurs when two or more species influence each other's evolutionary trajectory. For example, when a predator evolves new hunting tactics, prey species must evolve new defense mechanisms, leading to a back-and-forth evolutionary interaction. 5. Evolutionary implications of the Red Queen Hypothesis. The Red Queen Hypothesis has significant implications for understanding evolutionary dynamics. Constant evolutionary pressure. The hypothesis suggests that there is no ultimate end point to evolution. Organisms must keep evolving to survive, rather than evolving toward a perfect or optimal state. This provides an explanation for why some species appear to constantly evolve new traits, even when those traits do not provide a clear improvement in survival. Extinction and company evolution. Species that fail to adapt quickly enough to the evolutionary changes in other species face a higher risk of extinction. The Red Queen Hypothesis emphasizes that evolutionary stasis, i.e., the absence of change, is unlikely in an ecosystem with active coevolutionary processes. Sexual reproduction. The hypothesis has been used to explain the benefits of sexual reproduction. Environments where species are in constant evolutionary conflict, such as predator-prey or host-parasite interactions, sexual reproduction allows for genetic diversity, providing species with the ability to adapt rapidly to changes in their environment or interactions. 6. Evidence for the Red Queen Hypothesis. Evidence for the Red Queen Hypothesis comes from various biological and ecological studies. Host-parasite coevolution. Studies of host-parasite relationships, particularly between plants and pathogens or animals and parasites, support the Red Queen idea that species must constantly evolve to fend off evolving threats. For instance, the rapid evolution of resistance in agricultural plants to evolving pathogens has been observed. Sexual reproduction and genetic diversity. There is evidence that sexual reproduction, which generates genetic diversity, may be particularly advantageous in rapidly changing environments, consistent with the Red Queen Hypothesis. Sexual organisms may have an evolutionary advantage in the face of coevolutionary pressures. Predator-prey arms races. The ongoing arms race between predators and prey, as seen in species such as cheetahs and gazelles, supports the Red Queen model, with both species continually evolving in response to each other's adaptations. 7. Previous year gave questions on Red Queen Hypothesis Q1. Which of the following best illustrates the concept of the Red Queen Hypothesis? Evolutionary stasis in isolated populations. B. Predators and prey continuously evolving in response to each other. C. Symbiotic relationships between different species. C. A single species evolving in response to a changing environment. Answer. B. Predators and prey continuously evolving in response to each other. Explanation. The Red Queen hypothesis describes the continuous evolutionary arms race between species like predators and prey, where each species must constantly evolve to maintain their relative fitness. 2. According to the Red Queen hypothesis, evolutionary change in one species is independent of evolutionary changes in other species. B. Driven by genetic drift alone. C. A response to evolutionary changes in other species. D. Relevant to survival and reproduction. Answer. C. A response to evolutionary changes in other species. Explanation. The Red Queen hypothesis suggests that evolutionary changes in one species are often a direct response to the evolutionary changes in other species, as in the case of predator-prey or host-parasite interactions. 8. Conclusion. The Red Queen hypothesis offers a compelling explanation for the continual evolution of species in competitive environments. It highlights the importance of coevolution and the ongoing need for species to adapt in response to the changing dynamics within their ecosystems. The hypothesis underscores that evolutionary progress does not always equate to improvement or increased complexity, but rather to the maintenance of relative fitness in a world of constant ecological and evolutionary challenges. You said, co-speciation. ChatGPT said, ChatGPT company speciation. Co-speciation refers to the process in which two or more species undergo parallel speciation due to their close ecological relationship. In this phenomenon, when one species diversifies into multiple species, its associated counterpart species also diverges in tandem, often due to strong ecological dependencies or symbiotic relationships between them. This process is commonly observed in the interactions between hosts and their parasites, mutualistic relationships, or closely linked predator-prey species. Overview of co-speciation. Co-speciation occurs when two species are linked in such a way that changes in one species lead to changes in the other. It is an evolutionary process where speciation events in one species lead to speciation events in a closely associated species, typically because of close biological relationships such as parasitism, mutualism, or even predator-prey dynamics. In the case of host-parasite relationships, the evolution of one species often drives the evolutionary changes of the other. If the host species undergoes speciation due to environmental pressures or genetic divergence, the parasite species, which is often highly specialized to the host, may also undergo speciation to adapt to the new conditions of the host population. This process can lead to parallel evolutionary changes in both species, with each one influencing the other's speciation history. 2. Mechanisms of co-speciation. Several mechanisms underlie co-speciation, particularly in host-parasite, mutualistic, and predator-prey interactions. Host-parasite co-speciation. In parasitic relationships, the evolution of the host can directly impact the evolution of its parasites. For example, if a host species splits into two populations due to geographic or ecological isolation, its parasites may also diverge to adapt to the new host populations. Over time, this can lead to the formation of two new species of parasites that are genetically adapted to the two distinct populations of hosts. Symbiotic co-speciation. In mutualistic relationships, species evolve together due to the benefits they provide each other. For instance, certain species of flowering plants and their pollinators, e.g., bees, exhibit co-speciation. When the plant species undergoes speciation, the pollinators may also evolve into new species that are better adapted to the newly differentiated plants. Predator-prey co-speciation. In predator-prey dynamics, co-speciation can occur when predators and their prey evolve in response to each other's adaptations. For instance, prey may evolve better camouflage or defensive mechanisms, which in turn forces predators to evolve new hunting strategies. 3. Evidence for county speciation. Co-speciation has been widely documented in biological studies, particularly in parasitic systems. Parasite-host systems. One of the most well-documented examples of co-speciation occurs in the relationship between lice and their host mammals. When the host species splits into different populations, the lice that live on them tend to speciate as well, reflecting the divergence of their hosts. This co-speciation is supported by molecular evidence, where host and parasite phylogenies often show parallel branching patterns. Molecular evidence. Advances in molecular techniques, such as DNA sequencing, have provided significant evidence for co-speciation. Phylogenetic trees constructed for both hosts and parasites often show striking similarities in their branching patterns, suggesting that they have diverged together through co-speciation. Symbiotic relationships. Co-speciation is also evident in mutualistic relationships. For example, the co-evolution of coral species and their symbiotic algae, Zooxanthellae, has been shown to involve parallel diversification. Both coral and algae evolve in response to each other's ecological needs, resulting in co-speciation. 4. Factors influencing co-speciation. Several factors can influence the occurrence and extent of co-speciation. Ecological specialization. Species that are highly specialized to one another are more likely to undergo co-speciation. For example, highly host-specific parasites, such as lice or mites, are more likely to speciate when their host species diversifies, as they have a narrow ecological niche that limits their ability to switch to new hosts. Geographic isolation. Geographic barriers that cause isolation of populations can lead to genetic divergence and speciation. If one species in a co-evolutionary relationship is geographically isolated, it may undergo speciation, leading to speciation in the other species as well. Ecological dependencies. Strong ecological interactions, such as obligate mutualism or parasitism, increase the likelihood of co-speciation. For example, mutualistic relationships like those between certain plants and their pollinators, or between cleaner fish and their hosts, often lead to speciation events in both species. Phylogenetic congruence. The degree to which the evolutionary histories of two species mirror each other can provide evidence of co-speciation. If two species share a similar branching pattern on a phylogenetic tree, this is often a sign that they have evolved in tandem. 5. Applications of co-speciation. Conservation biology. Understanding co-speciation can be important for conservation efforts, especially when both host and parasite species are at risk of extinction. Protecting one species in a co-evolutionary pair may also indirectly protect the other species. Evolutionary biology. Co-speciation provides valuable insight into evolutionary processes, particularly in understanding how species evolve in response to their ecological interactions. It helps explain the interdependence of species in nature and how their evolutionary histories are interconnected. Genetics. The study of co-speciation plays a critical role in phylogenetic studies, especially in cases where the evolutionary history of a species can be traced through its relationship with another species, e.g., parasites and hosts. Molecular data from both the host and parasite species are often used to infer their co-evolutionary history. 6. Previous year questions on co-speciation. Q1. Co-speciation is most commonly observed in which of the following interactions? A. Predator-prey relationships. B. Competition between species. C. Mutualistic or parasitic relationships. D. Interspecific competition. Answer. C. Mutualistic or parasitic relationships. Explanation. Co-speciation is primarily observed in interactions where species are highly dependent on each other, such as mutualistic or parasitic relationship. For example, host-parasite co-speciation is a well-known instance. Q2. Which of the following would most likely lead to co-speciation? A. Predator evolving to catch faster prey. B. A parasite adapting to different host species. C. A mutualistic relationship between a flower and its pollinator. D. A plant evolving to resist herbivores. Answer. C. A mutualistic relationship between a flower and its pollinator. Explanation. In mutualistic relationships, such as those between plants and their pollinators, both species are often interdependent, which can lead to co-speciation as both evolve in response to each other. Q3. In a co-speciation event, the phylogenies of two species, host and parasite, are likely to A. Show random divergence. B. Reflect the same evolutionary patterns. C. Be unrelated. D. Diverge independently. Answer. B. Reflect the same evolutionary patterns. Explanation. In co-speciation, the evolutionary histories of host and parasite species are often parallel. When the host species diverges, the parasite species usually undergoes speciation as well, leading to similar phylogenetic branching patterns. 7. Conclusion. Co-speciation plays a crucial role in understanding the dynamics of species interactions, especially in ecosystems where species are tightly interdependent. Whether in parasitic, mutualistic, or other ecological interactions, co-speciation provides insights into the evolutionary process, showing how closely linked species evolve in parallel. This concept is fundamental in evolutionary biology and offers valuable perspectives on the interdependence of species in nature. It understands intricate relationships that drive speciation and diversification in ecosystems, highlighting the importance of evolutionary ecology and shaping the biodiversity of life on Earth. You said. Prey-predator interactions. ChatGPT said. ChatGPT. Prey-predator interactions. Prey interactions are central to understanding the dynamics of ecosystems and the evolutionary processes that shape species populations. In these interactions, predators exert selective pressures on prey populations, while prey species develop various adaptations to avoid being eaten. Over time, both predators and prey can co-evolve, leading to a dynamic, reciprocal relationship. These interactions are an example of negative feedback mechanisms in nature, where increases in predator populations can reduce prey populations, and conversely, a decline in prey can lead to a reduction in predator numbers, creating a balance in population sizes. 1. Overview of prey-predator interactions. Predator interactions can be studied in terms of both the behavioral and evolutionary aspects. In behavioral ecology, these interactions help explain how animals adapt their behaviors to maximize their survival and reproductive success in the presence of predators. On the evolutionary scale, they drive the process of natural selection, leading to the development of various adaptations in prey and predators alike. The primary characteristic of these interactions is that the predator benefits by consuming the prey, which provides the predator with energy and nutrients. Meanwhile, the prey is harmed by being eaten, which directly affects its survival and fitness. However, these interactions can also be mutually beneficial when prey and predator species share a more complex relationship, e.g., mutualistic relationships. 2. Types of predator-prey interactions. Predator-prey interactions can be classified based on the trophic level and nature of the interactions. True predation. In this interaction, the predator kills and consumes the prey. True predators, such as lions, carnivores preying on herbivores like antelopes, or hawks preying on smaller animals, directly benefit from the consumption of the prey. This is the most common type of interaction. Herbivora. While herbivores do not usually kill plants, they consume them. Plants may adapt through defensive mechanisms like thorns or toxins to reduce the impact of herbivora, leading to an evolutionary arms race between plants and herbivores. Parasitism. In parasitic interactions, the predator-parasite benefits at the expense of the host organism, but without necessarily killing it immediately. Parasites, such as ticks or intestinal worms, harm their hosts by feeding off their tissues or bodily fluids. Cannibalism. In some cases, individuals within the same species engage in cannibalism, where one individual preys on another. Cannibalism is often seen in extreme situations, such as when food is scarce or among species with high competition. 3. Evolutionary consequences of prey-predator interactions. The predator-prey relationship drives a variety of evolutionary outcomes. Adaptive evolution in prey. Prey species evolve to avoid or escape predators, often through adaptations like camouflage, warning coloration, aposematism, or the development of chemical defenses, e.g., toxins. Behavioral adaptations, such as herding or fleeing, also increase survival chances. Adaptive evolution in predators. Predators evolve specialized traits that allow them to catch and subdue prey. These may include sharp claws, teeth, or enhanced senses such as keen vision, smell, or hearing. Predators may also develop complex hunting strategies, such as cooperative hunting in wolves or stalking behavior in lions. Co-evolutionary arms race. As prey species evolve defenses to avoid predation, predators are simultaneously evolving better mechanisms to overcome those defenses. This creates a feedback loop of continuous adaptation, where the evolution of prey defenses prompts the evolution of predator offensive strategies, and vice versa. 4. Predator-prey models. One of the classic ways to model predator-prey interactions mathematically is through the Lotka-Volterra equations, which describe the dynamics of two interacting species, one predator and one prey. The Lotka-Volterra model consists of two equations. Prey population dynamics, D. NDT equals Rn minus ANP dt dN. Equals Rn minus ANP. N. Equals prey population size R. R equals intrinsic growth rate of the prey population P. P equals predator population size A. A equals predation rate coefficient. This equation indicates that the prey population grows exponentially in the absence of predators, represented by R n. Rn, but it decreases as predators consume prey, minus ANP minus ANP. Predator population dynamics, D. PDT equals BANP minus DPDT dP equals BANP minus dP. P equals predator population size A equals predation rate coefficient from the prey equation B equals efficiency of converting consumed prey into new predator offspring D equals mortality rate of the predator. This equation reflects that the predator population grows when they consume prey, but the growth is limited by the availability of prey and the mortality rate D. PDP. The Lacta-Volterra model demonstrates cyclical population dynamics. As predator numbers increase, prey numbers decrease, and as prey numbers decrease, predator numbers decline. This leads to oscillations in both predator and prey populations over time. 5. Functional responses of predators. Predators can exhibit different functional responses depending on how prey abundance affects their predation rate. There are three main types of functional responses. Type 1 functional response. The predator's rate of consumption increases linearly with prey density. This type of response is observed in simple predatory behaviors, where the predator can consume prey at a constant rate as prey numbers increase, until the predator reaches a saturation point. Type 2 functional response. In this case, the predator's consumption rate increases with prey density but at a decreasing rate. This response occurs when handling time, the time it takes to catch, kill, and consume a prey item, limits the predator's ability to consume prey. Prey density increases, the predator becomes more saturated and cannot eat faster than a certain rate. Type 3 functional response. Here, the predator's consumption rate increases in a sigmoidal curve. This response is common when predators switch to different prey types at low prey densities, but once prey density increases, the predator's rate of consumption rises sharply. 6. Defense mechanisms of prey. Prey species develop a variety of adaptations to escape, deter, or reduce the success of predators. Some of the most notable defense strategies include camouflage, cryptic coloration. Prey species evolve to blend into their environment, making it difficult for predators to spot them. This is seen in animals like the peppered moth or in the coloration of certain fish species. Warning coloration, aposematism. Some prey species develop bright, conspicuous colors that signal to predators that they are toxic or distasteful. Examples include poison dart frogs and certain species of butterflies. Mimicry. In mimicry, harmless prey species evolve to resemble harmful or dangerous species, fooling predators into avoiding them. An example is the viceroy butterfly, which resembles the toxic monarch butterfly. Chemical defenses. Some prey species produce toxins or other chemicals to deter predators. For example, many amphibians produce toxic secretions when threatened, and some plants produce toxic compounds to deter herbivores. Behavioral defenses. Prey may also employ behaviors that reduce predation risk, such as fleeing, hiding, or living in groups, e.g., herding an antelope. 7. Examples of prey-predator interactions. Lynx and snowshoe hare. One of the most well-known examples of predator-prey cycles is the relationship between the lynx and the snowshoe hare. The hare population increases, the lynx population also grows, as more prey is available. However, as the lynx population increases and consumes more hares, the hare population declines, leading to a decrease in lynx numbers, which allows the hare population to recover. Cheetahs and gazelles. Cheetahs hunt gazelles by sprinting at high speeds to catch them. Gazelles have evolved speed and agility as a defense against cheetah predation. The predator-prey interaction is a classic example of escape and pursuit dynamics. 8. Previous year gave questions on predator-prey interactions. Q1. In a predator-prey interaction, which of the following factors is most likely to affect the predator population size? A. Availability of water. B. Availability of prey. C. Prey's ability to escape. D. Predator's birth rate. Answer. B. Availability of prey. Explanation. The predator population size is directly influenced by the availability of prey, as predators rely on prey for food. If prey numbers increase, predators are more likely to increase in population as well. Q2. Which of the following represents the Lacta-Volterra model of predator-prey dynamics? A. The predator population size increases in response to prey population size, while the prey population size decreases as a result of predation. B. The prey population grows exponentially in the absence of predators. C. Both predator and prey populations reach equilibrium and remain constant. D. The predator population is independent of the prey population size. Answer. A. The predator population size increases in response to prey population size, while the prey population size decreases as a result of predation. Explanation. The Lacta-Volterra model shows cyclical oscillations between predator and prey populations, where predator numbers increase as prey are abundant, and prey numbers decrease as they are consumed by predators. 9. Conclusion. Predator interactions are a cornerstone of ecological dynamics and evolutionary processes. These interactions drive adaptive evolution in both predators and prey, shaping species behavior, morphology, and survival strategies. Understanding the complexity of these relationships is key to grasping the broader ecological systems and evolutionary mechanisms that govern diversity and population dynamics across ecosystems. You said, mimicry. ChatGPT said, ChatGPT. Mimicry in ecology and evolution. Mimicry is an evolutionary phenomenon where one organism, the mimic, resembles another organism, the model, in some way, often for the purpose of gaining a survival advantage. Mimicry can be broadly defined as an adaptive trait that helps the mimic avoid predation, gain resources, or achieve some other evolutionary benefit. Mimicry is a critical aspect of evolutionary biology, providing key insights into how natural selection shapes species interactions. Mimicry can be categorized into different types based on the nature of the relationship between the mimic and the model. The concept of mimicry involves a fitness benefit for the mimic, which arises from its resemblance to another organism or feature. 1. Types of mimicry. There are several well-known types of mimicry, each with its own specific ecological and evolutionary significance. These types can be classified primarily into Batesian mimicry, Müllerian mimicry, and aggressive mimicry. A. Batesian mimicry. Batesian mimicry is a form of mimicry in which a harmless species, the mimic, evolves to resemble a harmful or dangerous species, the model. The mimic benefits from the model's deterrent effects, such as avoiding predation because predators mistake the harmless mimic for a dangerous or unpalatable species. Key characteristics of Batesian mimicry. The mimic is harmless or palatable. The model is harmful, toxic, or dangerous, e.g., poisonous, aggressive. Predators avoid the mimic because they mistake it for the model. Example of Batesian mimicry. The viceroy butterfly, Limonitis archippus, resembles the monarch butterfly, Danaus plexippus. The monarch is toxic due to its diet of milkweed, which contains toxic compounds, while the viceroy is not toxic. Predators, such as birds, learn to avoid the monarch due to its unpalatability and also avoid the viceroy because of its similar appearance. Evolutionary significance. The mimic benefits because it avoids predation through resemblance to a harmful species, while the model benefits by deterring predators who avoid eating it. However, the mimic relies on the abundance of the model to gain the benefit, as predators must associate the model's harmful traits with the mimic. B. Müllerian mimicry. Müllerian mimicry is a form of mimicry where two or more harmful or unpalatable species evolve to resemble each other. Both species benefit from the mimicry because predators that have learned to avoid one harmful species are more likely to avoid the other, thus reducing the predation pressure on all mimics. Key characteristics of Müllerian mimicry. Both the mimic and the model are harmful or unpalatable. The mimic and the model have a mutual evolutionary advantage because predators that have learned to avoid one species will avoid others that resemble it. Example of Müllerian mimicry. The bee and the yellow jacket, a type of wasp, both share black and yellow warning coloration. Both species can sting and are harmful to predators, leading to mutual reinforcement of predator avoidance. Predators that have been stung by one of these species will likely avoid others with similar coloration. Evolutionary significance. The shared appearance between harmful species helps reinforce predator avoidance, which accelerates the learning process for predators. This form of mimicry is typically beneficial for all involved, especially when the mimics and models are coexisting in the same environment. See aggressive mimicry. Aggressive mimicry is a form of mimicry in which a predator or parasite mimics a harmless or beneficial organism in order to deceive its prey or host. The mimic's resemblance to a harmless or beneficial species allows it to approach its target without being detected or avoided, thus improving its chances of success in predation or parasitism. Key characteristics of aggressive mimicry. The mimic gains an advantage by resembling a species that is not feared or is beneficial to the target. The mimic uses this resemblance to deceive its prey or host into being consumed or parasitized. Example of aggressive mimicry. Anglerfish use a lure, a modified dorsal fin, that resembles a small fish or worm to attract prey. The prey approaches the lure, thinking it is food, only to be eaten by the anglerfish. Cuckoo birds, e.g., common cuckoo, Cuculus canorus, are known for laying their eggs in the nests of other bird species. The cuckoo chicks often mimic the begging calls of the host's own chicks, tricking the host parents into feeding them rather than their own offspring. Evolutionary significance. Aggressive mimicry provides a survival advantage to predators or parasites by allowing them to deceive their prey or hosts. This kind of mimicry demonstrates how species can exploit other species' behaviors or characteristics to their advantage. 2. Other forms of mimicry. While Batesian, Müllerian, and aggressive mimicry are the most well-known, other forms of mimicry exist. These include D. Automimicry, intraspecific mimicry. Automimicry refers to a mimicry that occurs within the same species, where one part of the organism resembles another part of the same organism to confuse predators or enhance survival. Example of automimicry. The false eye spot on the tail of certain butterflies like the peacock butterfly, Anacus resembles a large eye. This spot is thought to confuse or scare predators into attacking the tail rather than the head. E. Sexual mimicry. In sexual mimicry, one species mimics the sexual signals of another species to gain an advantage in terms of reproduction. This often involves males mimicking the appearance or behavior of females to reduce aggression from dominant males or increase mating opportunities. Example of sexual mimicry. In certain species of mimic spiders, male spiders mimic the mating signals of female spiders in order to avoid being eaten by the female, thereby increasing their chances of mating successfully. 3. Evolutionary significance of mimicry. Mimicry plays a critical role in the evolution of species by influencing the survival and reproductive success of organisms. The main evolutionary advantages of mimicry include reduced predation. By resembling harmful or unpalatable species, mimics can avoid being eaten. Enhanced survival. Mimics may benefit from mutualistic relationships with other species through mutual mimicry, which promotes cooperative survival. Increased reproductive success. Some forms of mimicry help organisms gain access to mates or improve their chances of offspring survival. 4. Previous year gave questions on mimicry. Q1. Which of the following is an example of Batesian mimicry? Resembling monarch butterfly, bee and wasp having similar coloration. C. Predator resembling a non-predator. D. Male mimic spiders resembling females to avoid predation. Answer. A viceroy butterfly resembling monarch butterfly. Batesian mimicry occurs when a harmless species, viceroy, resembles a harmful species, monarch, to avoid predation. Q2. One species resembling another to avoid predation. B. Two harmful or unpalatable species resembling each other. C. A predator mimicking a harmless species to deceive prey. D. A harmless species resembling a beneficial species. Answer. B. Two harmful or unpalatable species resembling each other. Explanation. In Müllerian mimicry, both species are harmful or unpalatable and evolve to resemble each other, reinforcing predator avoidance. Q3. What is the primary benefit of aggressive mimicry? By resembling harmful species. B. Deceiving prey or hosts into becoming a food source. C. Increasing reproductive success through sexual mimicry. D. Protecting the mimic from predators by resembling another species. Answer. B. Deceiving prey or hosts into becoming a food source. Explanation. Aggressive mimicry involves predators or parasites mimicking harmless or beneficial organisms to deceive their prey or host, increasing their chances of success. 5. Conclusion. Fascinating and diverse phenomenon in evolutionary biology, highlighting how species adapt to their environments to maximize their survival and reproductive success. Whether avoiding predators, gaining resources, or enhancing mating opportunities, mimicry showcases the power of natural selection in shaping the traits of organisms over time. Understanding the different types of mimicry, their evolutionary significance, and real-world examples provides valuable insight into the complex interactions that define ecological systems. Used. Crypsis ChatGPT said. ChatGPT. Crypsis, an evolutionary adaptation for concealment. Crypsis is an adaptive strategy where organisms evolve traits that help them blend into their environment, making them less detectable to predators or prey. It is a form of camouflage that involves hiding, blending in, or being cryptic, allowing organisms to avoid predation or increase their hunting success. Crypsis plays a survival and reproductive success of many species, and its evolution provides a fascinating example of how natural selection operates in the wild. 1. Types of crypsis. Crypsis can be categorized based on the type of camouflage an organism uses to avoid detection. The main types of crypsis are a background matching. Background matching is the most common form of crypsis, where an organism's appearance closely resembles the background or environment in which it lives. This type of camouflage makes it hard for predators to distinguish the organism from its surroundings. Example. Peppered moth Biston beetleria is a classic example of background matching. The moth has two main color morphs, one dark melanic and one light. During the Industrial Revolution, the dark morph became more common in polluted areas where tree bark was darkened by soot, as it blended better into the environment, making it less visible to predators. Evolutionary significance. Organisms with background matching have a higher chance of surviving predation, as their appearance makes them difficult to detect. The more closely an organism resembles its environment, the greater its chances of avoid detection. B. Disruptive coloration. Disruptive coloration is a form of camouflage where an organism has patterns or markings, such as spots, stripes, or patches that break up its outline or shape. These patterns confuse predators by making it harder to recognize the shape or silhouette of the organism. Example. The zebra Equus zebra is a good example of disruptive coloration. Its black and white stripes help break up its outline, making it difficult for predators to focus on a single part of its body. The stripes may also confuse predators, especially when zebras are in groups. Evolutionary significance. Disruptive coloration can help organisms avoid predation by making it harder for predators to detect and focus on them. In some cases, it can also confuse predators when an organism is in motion, making it harder to track. See countershading. Countershading involves having darker colors on the upper parts of the body and lighter colors on the lower parts. This creates the illusion of a uniform light source, which helps the organism blend into its surroundings and appear less three-dimensional. Example. Many animals, such as sharks, deer, and frogs, exhibit countershading. The darker top and lighter underside help these animals appear flat and blend into the surrounding environment, whether from above or below. For example, sharks' darker tops help them blend into the ocean depths when viewed from above, while their lighter undersides make them less visible when viewed from below against the lighter sky. Evolutionary significance. Countershading helps organisms avoid detection from predators, whether they are looking down from above or up from below. It effectively reduces the animal's visibility, making it harder for predators to locate and attack. Mimicry is a form of crypsis. Some organisms use mimicry as a form of crypsis to resemble inanimate objects, other species, or parts of their environment. This allows them to blend in more effectively and avoid detection. Example. The stick insect order Phasmatodea mimics twigs and branches, while leaf insects genus Phyllium resemble leaves. Insects remain motionless and are difficult for predators to detect because they look like parts of plants. Evolutionary significance. Mimicry allows organisms to avoid predation by resembling elements of the environment or other species that predators are less likely to attack. The more convincing the mimicry, the higher the chance of survival for the organism. Self-camouflage, adaptive color change. Some animals can actively change their color or appearance to match their surroundings. This form of crypsis provides flexibility for organisms that move between different habitats or environments. Example. The chameleon family Chameleonidae is well known for its ability to change its skin color to match its environment. Similarly, cuttlefish order Sepiota can change both the color and texture of their skin to blend into different backgrounds, such as coral reefs or rocky seabeds. Evolutionary significance. Self-camouflage provides a dynamic form of crypsis that can be employed in real time. This adaptation allows animals to move between environments with varying levels of concealment, increasing their chances of avoiding predators or surprising prey. 2. Evolutionary significance of crypsis. Crypsis offers several survival advantages, primarily by reducing the likelihood of being detected by predators. The main evolutionary benefits of crypsis include increased survival. By blending into the environment, organisms reduce their chances of being eaten by predators, thus enhancing their chances of survival. Increased reproductive success. Survivors are more likely to reproduce and pass on their cryptic traits to offspring, ensuring the persistence of these beneficial adaptations. Better hunting success. Predators that use crypsis can also increase their hunting success by remaining undetected by prey. Crypsis can also play a role in mate selection, as some species use camouflage to avoid competitors or to stay undetected during mating rituals. 3. Previous gave questions on crypsis Q1. Which of the following is an example of crypsis in animals? A. The viceroy butterfly mimicking the monarch butterfly. B. A chameleon changing its skin color to match the surroundings. C. A lion hunting in groups. D. Male peacocks displaying elaborate feathers to attract females. Answer. B. A chameleon changing its skin color to match the surroundings. Explanation. Crypsis involves avoiding detection, and the chameleon's ability to change its color to blend into its environment is an example of this adaptation. Q2. Disruptive coloration is most effective in which of the following contexts? A. Hiding from predators by blending into the background. B. Disrupting the shape and outline of the body to confuse predators. C. Mimicking the appearance of harmful species. D. Camouflaging in different habitats by changing color. Answer. B. Disrupting the shape and outline of the body to confuse predators. Explanation. Disruptive coloration breaks up the outline of the organism, making it harder for predators to identify and target specific parts of the body. Q3. Which of the following is an example of background matching? A. Zebra's black and white stripes blending into the tall grass. B. A polar bear's white fur blending into the snow. C. A frog mimicking the leaves on which it sits. D. A lion hunting in tall grass using stealth. Answer. B. A polar bear's white fur blending into the snow. Explanation. Background matching occurs when an organism's appearance closely matches the environment, making it less detectable. A polar bear's white fur helps it blend into snowy environments, evading predators or prey. 4. Conclusion. Crypsis is a vital evolutionary adaptation that enhances the survival of organisms by enabling them to avoid detection by predators or prey. Whether through background matching, disruptive coloration, countershading, mimicry, or self-camouflage, Crypsis provides numerous evolutionary advantages that help species thrive in their environments. Understanding the different forms and functions of Crypsis is essential to grasping the complexity of survival strategies in the natural world. You said. Crypsis, etc. Some more like these ChatGPT said. ChatGPT mimicry. Mimicry is an evolutionary adaptation where one organism, the mimic, closely resembles another organism, the model, or an object in its environment. This resemblance provides a survival advantage by deceiving predators, prey, or potential mates. There are several types of mimicry, and each serves different ecological purposes. Mimicry often plays a role in predator-prey interactions, competition, and even reproduction. 1. Types of mimicry. Batesian mimicry. Batesian mimicry occurs when a harmless or palatable organism, the mimic, resembles a harmful or unpalatable organism, the model. This adaptation helps the mimic avoid predation, as predators avoid the harmful model species. Example. Viceroy butterfly, Limonitis archippus, is a classic example. It mimics the toxic monarch butterfly, Danaus plexippus, which is distasteful to birds. Predators that have had negative experiences with monarchs will avoid the viceroy, even though it is not toxic. Evolutionary significance. The mimic gains protection from predators by resembling a dangerous species. For the model species, this form of mimicry is beneficial because it maintains its harmful characteristics, but it also helps the mimic avoid being preyed upon. B. Malarian mimicry. Malarian mimicry occurs when two or more harmful or unpalatable species resemble each other. In this case, both species benefit because predators learn more quickly to avoid them. The more often predators encounter harmful species with similar markings, the faster they will learn to avoid them. Example. Several species of bees and wasps exhibit Malarian mimicry. All these species share similar yellow and black coloration, signaling to predators that they are capable of stinging and should be avoided. Evolutionary significance. The mutual resemblance of harmful species reinforces the avoidance behavior of predators, which helps all species involved. It is the individual learning cost for predators, thus benefiting both the mimic and the model species. See aggressive mimicry. In aggressive mimicry, predators or parasites resemble a harmless or beneficial species to deceive their prey or hosts. This mimicry strategy helps the predator or parasite gain access to its victim more easily. Example. Anglerfish family Laphiidae is a notable example. They have a lure, a modified dorsal spine, that mimics a small prey item. Prey such as fish or smaller marine creatures approach the lure, thinking it is food, only to become prey themselves. Evolutionary significance. Aggressive mimicry helps predators or parasites increase their hunting efficiency by deceiving their prey into approaching, thereby increasing their chances of capturing a meal or host. D. Automimicry, intraspecific mimicry. Automimicry, or interspecific mimicry, occurs when an organism mimics a part of its own body to gain a survival advantage, often in the context of defense mechanisms. Example. Snakes such as the eastern coral snake, Micrurus fulvius, exhibit interspecific mimicry by having a pattern that resembles their own tail at the front of their bodies, confusing predators, and predators may hesitate before attacking. Evolutionary significance. Automimicry can help reduce predation risks by confusing predators about the animal's vulnerable parts. It may also play a role in deterring rivals or attracting mates. 2. Evolutionary significance of mimicry. Mimicry has important implications for both the mimics and the models in evolutionary terms. Protection. Mimicry helps the mimic avoid being eaten or harmed by predators. It allows the mimic to exploit the model's defenses or reputation for safety. Increased fitness. Mimicry can help the mimic survive and reproduce, leading to an increase in the mimic's fitness by passing on the beneficial traits. Coevolution. The relationship between mimics and models often leads to coevolutionary dynamics. Models may evolve more complex or refined warning signals, while mimics may evolve to become even more convincing in their resemblance. 3. Previous year gave questions on mimicry. Q1. What is Batesian mimicry? Both the mimic and the model are harmless. B. When a harmless organism mimics a harmful or toxic organism. C. When both the mimic and the model are harmful. D. When an organism mimics an inanimate object. Answer. B. When a harmless organism mimics a harmful or toxic organism. Explanation. Batesian mimicry involves a harmless organism resembling a harmful or toxic one, thereby avoiding predation. Q2. In Malarian mimicry, which of the following is true? A. The mimic benefits by appearing harmful, and the model also benefits from being dangerous. B. The mimic benefits by resembling a harmless species, and the model gains from being unpalatable. C. Both mimic and model are harmless. D. The mimic and model do not share similar appearance. Answer. A. The mimic benefits by appearing harmful, and the model also benefits from being dangerous. Explanation. In Malarian mimicry, both the mimic and the model share similar warning signals, and they are both unpalatable or harmful to predators, benefiting both species. Q3. Which of the following is an example of aggressive mimicry? A. Viceroy butterfly mimicking a monarch butterfly. B. An anglerfish using a lure to attract prey. C. A stick insect mimicking twigs. D. A cuckoo bird laying eggs in another bird's nest. Answer. B. An anglerfish using a lure to attract prey. Explanation. Aggressive mimicry involves a predator mimicking a harmless or attractive object to lure prey, as seen in the anglerfish's use of a lure to attract smaller fish. 4. Conclusion. Mimicry is an essential evolutionary strategy that increases the survival and reproductive success of organisms by either avoiding predators or enhancing hunting abilities. It highlights the complex interactions between species and the role of natural selection in shaping these fascinating survival strategies. The types of mimicry, Batesian, Malarian, aggressive, and automimicry, each demonstrate unique ways organisms can evolve to avoid threats or exploit opportunities in their environments. Crypsis and mimicry, comparison. Both crypsis and mimicry are camouflage strategies that help organisms avoid detection, but they differ in how they achieve this goal. Crypsis involves blending into the environment through background matching, disruptive coloration, or countershading to avoid detection. Mimicry, on the other hand, involves one species resembling another species or object to gain a survival advantage, such as by tricking predators or prey. Both strategies represent the incredible diversity of adaptations in the natural world, shaped by millions of years of evolutionary pressures. You said, related topics. ChatGPT said, ChatGPT. Here are some related topics that connect well with crypsis and mimicry, which are crucial concepts in evolutionary ecology. Understanding these concepts helps in getting a comprehensive view of the diverse strategies organisms use to survive and thrive in their respective environments. 1. Camouflage. General concept. Camouflage is the broader category that includes both crypsis and mimicry. It is any adaptation that helps an organism avoid detection by blending into its environment or mimicking other organisms or objects. Camouflage plays a crucial role in avoiding predators or prey species or enhancing hunting efficiency for predators. Crypsis involves blending into the environment, making the organism less visible. Mimicry involves resembling another organism or object to avoid detection or capture. Example, the leaf-tailed gecko Uroplatus from Madagascar resembles dead leaves and tree bark. It uses camouflage as a form of crypsis to avoid detection from predators. 2. Anti-predator adaptations. These are strategies developed by prey organisms to reduce the likelihood of being captured by predators. Anti-predator adaptations include cryptic coloration. Crypsis, as discussed, organisms blend into their environment. Warning coloration. Bright, conspicuous colors signal to predators that an organism is toxic or dangerous, e.g., the bright colors of a poison dart frog. Behavioral adaptations. Some species may freeze, flee, or exhibit misleading behaviors, e.g., playing dead like opossums. Defensive mechanisms. Many animals have evolved physical defenses like spines, e.g., porcupines, hard shells, e.g., tortoises, or venom, e.g., snakes. Example, the poison dart frog uses warning coloration to signal to predators that it is toxic, reducing the likelihood of being eaten. 3. Evolutionary arms race. The evolutionary arms race describes the ongoing struggle between predators and prey or parasites and hosts, where each side evolves counter-adaptations to outcompete the other. Prey defenses, e.g., crypsis, mimicry, or chemical defenses. Predators evolve better strategies to overcome these defenses, e.g., better eyesight to detect camouflage prey or chemical resistance. Example, the arms race between cheetahs and gazelles. Cheetahs evolve faster running speeds to catch prey, while gazelles evolve evasive maneuvers and agility to escape. 4. Warning coloration. Aposematism. Aposematism is a form of mimicry where a species evolves bright, warning colors or markings to signal to potential predators that it is toxic, venomous, or otherwise dangerous. This is a mutualistic strategy for both the mimic and the model. Aposematism is most often observed in insects and amphibians. It works because predators learn to avoid these brightly colored organisms after a few negative encounters. Example, the monarch butterfly and viceroy butterfly. Monarchs are toxic due to chemicals in their bodies, while viceroys mimic their appearance to gain protection from predators, despite being harmless. 5. Eusociality and cooperative defense. Eusociality is a social structure where individuals of a species cooperate for the good of the group, often to defend against predators. It is most commonly found in insects like ants, bees, and termites. Alarm calling. Certain individuals within a colony may act as sentinels to warn others of approaching predators. Group defense. Some species, like meerkats or ants, engage in cooperative defense, where the group actively protects its members. Example, African honeybees exhibit eusociality, where worker bees protect the queen and the hive from predators, using a collective defense strategy. 6. Predator adaptations for detection. Predators, too, have evolved a variety of adaptations to overcome the defenses of their prey, such as crypsis or mimicry. Improved vision. Some predators have evolved better eyesight, such as ultraviolet vision, e.g., birds of prey. Detection. Predators may have a heightened sense of smell or taste to detect hidden prey, e.g., bloodhounds or certain species of sharks. Mimicry of prey. Some predators mimic the appearance or behavior of their prey's food source or habitat, aiding in ambush predation, e.g., anglerfish's lure. 7. Evolution of coloration. Coloration in animals can evolve for various reasons, not just for crypsis or mimicry, but also for thermoregulation, social signaling, and mating displays. Melanism, the increased presence of dark pigments in the skin, scales, or fur of an animal. Albinism, the absence of pigmentation, which can make an organism highly visible to predators, although in some cases, it may provide benefits in specific environments. Coloration in birds. Bright colors in birds may not only be used for camouflage but also for attracting mates or signaling aggression. Example, peppered moth Biston beetleria evolved darker coloration in industrial areas during the Industrial Revolution due to the prevalence of soot-covered trees. 8. Parasite mimicry and host manipulation. Some parasites evolve mimicry strategies to manipulate their hosts into behaving in ways that benefit the parasite's survival and reproduction. This often involves changing the appearance or behavior of the host to ensure the parasite's continued life cycle. Parasitoid wasps lay their eggs inside other insects, and their larvae can control the behavior of their hosts to protect them during their developmental stages. Parasitic plants like mistletoe mimic the host plant's tissues to infiltrate and extract nutrients without being detected. Example, cuckoo birds lay their eggs in the nests of other birds, mimicking the host bird's eggs to trick them into incubating and caring for the cuckoo chick. 9. Evolution of sexual dimorphism. Sexual dimorphism refers to the differences in size, coloration, shape, or behavior between males and females of the same species, typically due to sexual selection. In some species, males evolve vibrant colors or impressive features to attract mates, while in others, females may evolve traits that are more cryptic for hiding or nurturing young. Coloration. Male birds often have bright plumage to attract females. Size. Males may be larger or have stronger features to compete with other males for mates. Behavior. Males may perform elaborate mating displays or songs to signal their fitness to females. Example, the peacock Pavo cristatus exhibits sexual dimorphism, with males possessing large, colorful tails that they use in courtship displays to attract females. 10. Evolution of plant defenses. Plants have evolved various defenses to avoid being eaten by herbivores. These defenses include both physical and chemical strategies, and they often work in conjunction with mimicry or crypsis. Thorns and spines. These act as mechanical defenses to deter herbivores. Chemical defenses. Some plants produce toxic or distasteful chemicals to deter herbivores, e.g., alkaloids, phenols. In plants. Some plants mimic the appearance of animals or other dangerous species to avoid being eaten. Example, the passion flower passiflora uses large, brightly colored structures that resemble eyes to intimidate herbivores, mimicking the appearance of predators like snakes. Conclusion. The concepts of crypsis, mimicry, and camouflage form a central part of evolutionary theory, helping explain how species adapt to their environments in complex ways. By studying these topics, one can appreciate the intricate relationships between predators, prey, and the environments they inhabit. Evolution has shaped these strategies to enhance survival and reproduction, demonstrating the creativity of natural selection. You said. All terms related to this kind of interactions, ChatGPT said. ChatGPT. Here's a comprehensive list of terms related to interactions in ecology and evolution, specifically concerning crypsis, mimicry, and other forms of defense, signaling, and co-evolution between species. 1. Camouflage. Definition: the ability of an organism to blend into its environment to avoid detection by predators or prey. Related terms. Background matching: organisms resemble the environment in color or texture. Disruptive coloration: bold patterns that break up an organism's outline. Countershading: a form of shading where the upper body is darker than the underside to provide depth and reduce visibility. 2. Crypsis. Definition: a form of camouflage where organisms hide by blending into their surroundings. Related terms. Hide-and-seek behavior: behavioral changes used by animals to seek or hide. Mimicry: resembling an object or another organism to avoid predation. Behavioral crypsis: behavioral strategies used by organisms, like remaining still or avoiding open areas. Physiological crypsis: the ability of an organism to change color or texture to match the environment. 3. Mimicry. Definition: when one organism resembles another species or an object to gain a survival advantage. Types. Batesian mimicry: harmless species mimic harmful or toxic species to avoid predators. Malarian mimicry: two harmful or unpalatable species resemble each other to reinforce avoidance behavior in predators. Two species that resemble one another to amplify their protective signal to predators. 4. Aposematism: warning coloration. Definition: bright coloration or warning signals used by toxic or dangerous species to deter potential predators. Related terms. Conspicuous coloration: bright, visible color patterns signaling danger. Toxicity: the harmful chemicals that some organisms possess to deter predators. Chemical defenses: production of toxins, venom, or other chemical deterrents. 5. Anti-predator strategies. Definition: adaptations developed by prey to avoid being caught by predators. Related terms. Escape behaviors: flight, rapid movement, or camouflage to evade predators. Predator confusion: behavioral or visual tactics, such as rapid movement or sudden changes in direction, to confuse predators. Mobbing: cooperative behavior among prey to confuse or drive away predators. Defensive postures: behaviors or stances adopted by prey to deter predators, e.g., erecting spines, inflating bodies. Alarm calling: vocal signals or other signs given by prey to alert others of a predator's presence. 6. Evolutionary arms race. Definition: the ongoing, dynamic evolutionary process in which predators and prey, or parasites and hosts, evolve in response to each other's adaptations. Related terms. Co-evolution: reciprocal evolutionary change between two or more interacting species. Red Queen hypothesis: the idea that species must continuously evolve to maintain their relative fitness, as their competitors also evolve. Host-parasite co-evolution: the back-and-forth evolutionary adaptations between parasites and their hosts. Predator-prey co-evolution: the evolution of traits in predators and prey that drive mutual evolutionary change. 7. Sexual selection. Definition: a form of natural selection where traits that increase an individual's chances of reproducing are favored, often leading to sexual dimorphism. Related terms. Mate choice: the selection of a mate based on desirable traits. Sexual dimorphism: differences in size, shape, or coloration between males and females of the same species. Courtship displays: behavioral displays by males to attract females, e.g., peacock tail feathers. Sexual conflict: when the reproductive interests of males and females are in opposition. Sperm competition: a form of sexual selection in which male reproductive success is determined by the outcome of competition between sperm from different males. 8. Predator-prey interactions. Definition: the dynamic interactions between predators and their prey, influencing the evolution of both species. Related terms. Predator avoidance: adaptations in prey that allow them to avoid being captured, e.g., speed, crypsis, mimicry. Functional response: the relationship between prey density and predator's rate of consumption. Numerical response: the change in predator population size in response to the availability of prey. Optimal foraging theory: the theory that animals forage in such a way as to maximize the net energy gain per unit of time spent foraging. 9. Co-evolution. Definition: the process in which two or more species exert selective pressures on each other, leading to reciprocal evolutionary changes. Related terms. Mutualism: a type of co-evolution where both species benefit from the interaction, e.g., pollination between flowers and bees. Antagonistic co-evolution: when one species evolves traits that harm the other species, prompting a reciprocal defensive adaptation. Symbiosis: a broad term that includes all types of close interactions between species, including mutualism, parasitism, and commensalism. Co-speciation: the process by which two species evolve in response to each other and speciate in tandem, often seen in parasites and their hosts. 10. Disruptive selection. Definition: a form of natural selection where extreme values for a trait are favored over intermediate values, leading to the divergence of two distinct phenotypes. Related terms. Bimodal distribution: a pattern of variation in a population where two distinct forms are present, often due to disruptive selection. Speciation: the formation of new and distinct species due to reproductive isolation or ecological divergence. Diversifying selection: another term for disruptive selection, where two or more phenotypic extremes are favored. 11. Genetic drift. Definition: a random process that causes allele frequencies to change over time, particularly in small populations. Antagonistic co-evolution: when one species evolves traits that harm the other species, prompting a reciprocal defensive adaptation. Symbiosis: a broad term that includes all types of close interactions between species, including mutualism, parasitism, and commensalism. Co-speciation: the process by which two species evolve in response to each other and speciate in tandem, often seen in parasites and their hosts. 10. Disruptive selection. Definition: a form of natural selection where extreme values for a trait are favored over intermediate values, leading to the divergence of two distinct phenotypes. Related terms. Bimodal distribution: a pattern of variation in a population where two distinct forms are present, often due to disruptive selection. Speciation: the formation of new and distinct species due to reproductive isolation or ecological divergence. Diversifying selection: another term for disruptive selection, where two or more phenotypic extremes are favored. 11. Genetic drift. Definition: a random process that causes allele frequencies to change over time, particularly in small populations. Related terms. Founder effect: a form of genetic drift that occurs when a small group of individuals establishes a new population. Bottleneck effect: a sharp reduction in the size of a population due to environmental events, leading to a loss of genetic diversity. Random mating: the assumption that individuals pair randomly, which can influence allele frequencies in populations. 12. Mutualism. Definition: a type of coevolution where both interacting species benefit from the relationship. Related terms. Pollination: a form of mutualism where pollinators, e.g., bees, birds transfer pollen between flowers, aiding in plant reproduction. Seed dispersal: a mutualistic interaction in which animals transport seeds, aiding in plant reproduction while benefiting from the fruit. Cleaning symbiosis: a relationship in which cleaner organisms, e.g., cleaner fish, remove parasites from host organisms, e.g., large fish. 13. Evolutionary ecology. Definition: the study of the interactions between ecological processes and evolutionary change. Related terms. Adaptation: the process by which an organism becomes better suited to its environment through natural selection. Fitness: the ability of an organism to survive, reproduce, and pass on its genes to the next generation. Niche partitioning: the process by which competing species divide resources to minimize competition and coexist. 14. Altruism. Definition: a behavior where an organism acts in a way that benefits others at a cost to itself. Related terms. Kin selection: a form of natural selection where organisms increase the survival of their relatives, even at the expense of their own reproduction. Reciprocal altruism: a behavior where organisms help each other with the expectation of receiving help in return in the future. Conclusion. The terms provided above cover a range of concepts related to interactions in ecology and evolution. These interactions form the foundation of understanding evolutionary strategies, behavioral adaptations, and ecological dynamics. Familiarity with these terms will help you grasp the complexities of how organisms evolve and adapt in response to environmental pressures, species interactions, and the ongoing processes of natural selection and coevolution. You said. Population and quantitative genetics. ChatGPT said. ChatGPT. Population and quantitative genetics. Population and quantitative genetics are key fields within evolutionary biology that focus on understanding the genetic structure of populations and how genetic traits vary and evolve within these populations. These fields involve studying gene frequencies, genetic variation, and the forces that drive evolutionary changes in populations. Here's a detailed breakdown of the topic. 1. Population genetics definition. Population genetics is the study of genetic variation within populations and how evolutionary forces such as natural selection, genetic drift, mutation, and gene flow influence this variation over time. Key concepts in population genetics: gene pool. The total collection of alleles in a population at a given time. Gene flow and mutation can alter the gene pool by introducing new alleles or changing allele frequencies. Allele frequency. The proportion of a particular allele among all allele copies in a population. It is an essential concept in measuring genetic variation. Hardy-Weinberg equilibrium. A mathematical model used to measure allele and genotype frequencies in a population that is not evolving. The 5 conditions for Hardy-Weinberg equilibrium are: no mutation, random mating, no natural selection, large population size, no genetic drift, no gene flow, immigration, emigration. Hardy-Weinberg equation. P2 plus 2PQ plus Q2 equals 1 P2. Plus 2PQ plus Q2 equals 1 where P equals frequency of dominant allele Q. Q equals frequency of recessive allele P. 2P2 equals frequency of homozygous dominant genotype 2. PQ. 2PQ equals frequency of heterozygous genotype Q. 2Q2 equals frequency of homozygous recessive genotype. Factors that affect allele frequencies. Natural selection favors certain alleles over others based on survival and reproduction. Genetic drift, random changes in allele frequencies, more pronounced in small populations. Mutation, the introduction of new alleles into the gene pool. Gene flow, the movement of alleles between populations through migration. Key population genetics models. Wright-Fisher model describes how allele frequencies fluctuate in a population due to genetic drift. Bottleneck effect. A sharp reduction in the size of a population due to environmental events, leading to a loss of genetic variation. Founder effect. When a small group of individuals forms a new population, the allele frequencies of the new population may be significantly different from the original population. 2. Quantitative genetics. Definition. Quantitative genetics is the study of genetic variation for complex traits that are controlled by many genes, each contributing a small amount to the overall phenotype. These traits exhibit continuous variation, e.g., height, weight, and yield rather than discrete categories, e.g., Mendelian traits. Key concepts in quantitative genetics: polygenic traits. Traits controlled by 2 or more genes, each contributing a small amount to the overall phenotype. Example: human height, skin color, or crop yield. Heritability. The proportion of observed variation in a trait that can be attributed to genetic variation. Narrow sense heritability, h squared, the proportion of phenotypic variation due to additive genetic effects. H. 2 equals. Additive genetic variance total phenotypic variance h. 2 equals. Total phenotypic variance additive genetic variance. Broad sense heritability, h squared, the proportion of phenotypic variation due to genetic variance, including additive, dominance, and epistatic variances. Phenotypic variation. The observable variation in a trait in a population, which is the result of both genetic and environmental factors. Phenotypic variance is partitioned into genetic and environmental components, v. P equals Vg plus V, Vp equals Vg plus Ve. Where V, PVP equals total phenotypic variance V. GVG equals genetic variance V. EVE equals environmental variance additive genetic variance V underscore A. The portion of genetic variance that is attributed to the additive effects of alleles. This component is the most important for selection, as it directly affects the response to selection. Genetic correlation. When 2 or more traits are genetically correlated, meaning they share a portion of their genetic variance. This can be due to pleiotropy, one gene affecting multiple traits, or linkage disequilibrium, non-random association of alleles at different loci. Selection response R. The change in the mean value of a trait in one generation due to natural or artificial selection. Breeder's equation. R equals H2 times S. R equals H2 times S where R equals response to selection H. 2H2 equals heritability S. S equals selection differential, the difference between the mean phenotype of selected individuals and the overall population mean 3. Practice questions and answers. Question 1, Gate 2018. What is the condition for Hardy-Weinberg equilibrium to hold in a population? Answer. The conditions for Hardy-Weinberg equilibrium to hold are: no mutation occurs, random mating occurs, no natural selection acts on the population, large population size, no genetic drift, no migration, gene flow occurs. Question 2, Gate 2020. In a population of frogs, the frequency of the dominant allele A is 0.6. What is the frequency of the heterozygous genotype A? Answer. Given that the frequency of the dominant allele P equals 0.6, P equals 0.6 and Q equals 1 minus P equals 0.4. Q equals 1 minus P equals 0.4. We can use the Hardy-Weinberg equation to calculate the frequency of the heterozygous genotype. 2PQ equals 2 times 0.6 times 0.4 equals 0.48. 2PQ equals 2 times 0.6 times 0.4 equals 0.48. So, the frequency of the heterozygous genotype A is 0.48. Question 3, Gate 2019. If a population has a heritability of 0.7 for a trait, and the selection differential S is 10, what is the response to selection R? Answer. Using the Breeder's equation R equals H2 times S, R equals H2 times S. R equals 0.7 times 10 equals 7. R equals 0.7 times 10 equals 7. So, the response to selection is 7. 4. Summary. Population and quantitative genetics are essential for understanding how genetic variation in populations evolves over time under the influence of evolutionary forces. Key concepts such as Hardy-Weinberg equilibrium, heritability, polygenic traits, and selection response provide the tools to analyze and predict evolutionary changes in populations. Studying these concepts in depth is crucial for fields such as conservation biology, breeding programs, and understanding the dynamics of natural populations. By mastering these topics, you will be equipped to tackle gate questions related to the genetic structure of populations, the evolution of quantitative traits, and the factors that influence genetic diversity and adaptation. You said. Origins of genetic variation, ChatGPT said. ChatGPT. Origins of genetic variation. Genetic variation is the raw material for evolution. It refers to differences in the genetic makeup genotype of individuals within a population, which contribute to variation in their phenotype's observable traits. Genetic variation is crucial for natural selection and the adaptability of species to changing environments. The origins of genetic variation are multifaceted and arise from several mechanisms, which are outlined below. 1. Mutations definition. A mutation is any change in the DNA sequence that can occur at different levels, ranging from a single nucleotide to large-scale chromosomal alterations. Mutations are the ultimate source of genetic variation in all populations. Types of mutations. Point mutations involve a change in a single nucleotide in the DNA sequence. Subtypes. Silent mutations, no change in amino acid sequence. Missense mutations result in a different amino acid being incorporated into the protein. Nonsense mutations lead to a premature stop codon, producing a truncated protein. Insertions and deletions, indels, involves the addition or loss of nucleotides in the DNA sequence, which can lead to frameshift mutations if the number of nucleotides added or deleted is not a multiple of 3. Chromosomal mutations. Changes in the structure or number of chromosomes, including deletions, duplications, inversions, and translocations. Causes of mutations. Spontaneous mutations occur naturally due to errors in DNA replication or repair mechanisms. Induced mutations caused by external factors such as radiation, chemicals, or viruses. Role in genetic variation. Mutations generate new alleles, some of which may provide a selective advantage or disadvantage. Mutations in germ cells, eggs or sperm, are passed on to offspring and contribute to genetic diversity in populations. 2. Genetic recombination. Definition. Genetic recombination is the process by which offspring inherit a combination of alleles from their parents that is different from those present in the parental genotypes. It occurs during meiosis and is a key mechanism for generating genetic variation. Mechanisms of genetic recombination crossing over. During meiosis, homologous chromosomes exchange genetic material at regions known as chiasmata. This results in new combinations of alleles on chromosomes. Independent assortment. The random distribution of homologous chromosomes to gametes during meiosis. Different combinations of maternal and paternal chromosomes are inherited by offspring, increasing genetic diversity. Gene conversion. A process where one allele replaces another during recombination, contributing to genetic variation, though it is a rare event. Role in genetic variation. Recombination reshuffles genetic material, producing new allele combinations that increase genetic diversity. The shuffling of alleles also facilitates the adaptive potential of populations by generating novel genotypes. 3. Gene flow, migration. Definition. Gene flow refers to the transfer of alleles between populations through migration or interbreeding. When individuals from different populations mate, they introduce new genetic material into the gene pool of the receiving population, which can alter allele frequencies. Mechanisms of gene flow. Immigration. The arrival of individuals from another population introduces new genetic material. Emigration. The departure of individuals removes alleles from the population, though the effect on genetic variation depends on the genetic composition of the emigrants. Role in genetic variation. Gene flow can increase genetic variation by introducing new alleles. In small populations or isolated populations, gene flow can reduce genetic differences between populations, leading to genetic homogeneity. 4. Genetic drift. Definition. Genetic drift is the random fluctuation of allele frequencies in a population due to chance events. It is most pronounced in small populations and can lead to the loss of alleles, including beneficial or neutral ones. Mechanisms of genetic drift. Bottleneck effect. A drastic reduction in population size due to an environmental event, leading to a loss of genetic variation. Founder effect. A small group of individuals from a population establishes a new population, and the genetic variation of the new population is limited to that of the founders. Role in genetic variation. Genetic drift can reduce genetic variation, especially in small populations. In some cases, genetic drift may lead to fixation, the complete disappearance of alleles in a population. In the long term, genetic drift can limit the evolutionary potential of populations by reducing diversity. 5. Sexual reproduction. Definition. Sexual reproduction involves the combination of genetic material from two parents, creating offspring with a unique combination of alleles. This process plays a significant role in maintaining and generating genetic variation in populations. Mechanisms involve mating. Random mating or non-random mating influences the genetic combinations that are passed on to the next generation. Outcrossing. The mating between genetically different individuals increases heterozygosity and genetic variation. Role in genetic variation. Sexual reproduction increases genetic variation by combining different alleles from two parents to create new combinations in offspring. It promotes genetic diversity through mechanisms such as recombination and independent assortment, which lead to varied genetic outcomes in the progeny. 6. Horizontal gene transfer, HGT. Definition. Horizontal gene transfer refers to the movement of genetic material between organisms other than through traditional reproduction, vertical gene transfer. This process is particularly important in bacteria and archaea but can also occur in other organisms. Mechanisms of HGT transformation. The uptake of naked DNA from the environment by a cell. Conjugation. The transfer of genetic material via direct cell-to-cell contact. Transduction. The transfer of genetic material via viruses, bacteriophages, that infect a cell and integrate viral DNA into the host genome. Role in genetic variation. HGT introduces new genes into a population and can rapidly increase genetic diversity, especially in prokaryotes. It plays an important role in the spread of advantageous traits such as antibiotic resistance. Practice questions and answers. Question 1, GATE 2017. Which of the following is the ultimate source of genetic variation in a population? A. Gene flow. B. Recombination. C. Mutation. D. Genetic drift. Answer. C. Mutation. Mutations are the ultimate source of genetic variation as they generate new alleles in the gene pool, whereas gene flow, recombination, and genetic drift are mechanisms that influence existing variation. Question 2, GATE 2019. Which of the following processes contributes to genetic variation in a sexually reproducing population? A. Independent assortment. B. Genetic drift. C. Mutation. D. Both and C. Answer. D. Both and C. Independent assortment during meiosis and mutations are two key processes that contribute to genetic variation in sexually reproducing populations. Question 3, GATE 2020. In a population of butterflies, the introduction of a new population with a different genetic background leads to a change in allele frequencies. This is an example of A. Genetic drift. B. Mutation. C. Gene flow. D. Natural selection. Answer. C. Gene flow. Gene flow occurs when individuals from different populations interbreed, introducing new genetic material and altering the allele frequencies of the receiving population. Summary. The origins of genetic variation are multifaceted and include mechanisms such as mutations, genetic recombination, gene flow, genetic drift, sexual reproduction, and horizontal gene transfer. These mechanisms interact to create the genetic diversity that forms the basis for evolution through natural selection and other evolutionary forces. Understanding these processes is crucial for explaining how populations evolve and adapt to changing environments, and it is an essential part of the GATE exam syllabus in evolutionary biology. You said, Mendelian genetics. ChatGPT said, ChatGPT. Mendelian genetics. Mendelian genetics refers to the principles of heredity discovered by Gregor Mendel in the 19th century, which form the foundation of classical genetics. Mendel's work focused on the inheritance of traits in pea plants and provided crucial insights into the way genes are passed from one generation to the next.